Okay, my friends, they're finally jumping on board here. The interstitium may be the largest organ in the body, making up 20% of a young adult's volume. It acts as a shock absorber for the body, protecting tissue in places that expand and contract. It's a really gooey substance. The interstitium may also explain how cancer spreads and where lymph comes from, the fluid that immune cells need to function. I've been talking about this for many years now. I wrote papers on fascia facilitated fossilization, which you'll see in great detail. And they say the discovery of interstitium has been controversial, and scientists continue to debate its implications. If other researchers agree the interstitium is an organ, anatomy textbooks may need to be revised. They need to absolutely be revised. Not only that, geology, because of mud fossils, Velikovsky's research, we need to get. These are all new books. They are from Paul Amatucci. Has them on Amazon. They're very inexpensive, and they're just from a lot of them are from my papers on the giants and on the electron flood theory and on the world. Is literally just the world. The world itself is basically made out of mud fossils. And now I find that the whole universe is. So we need new textbooks, absolutely. And we are going to have them. Things have to start over again, but we need to be able to look at these things with a rational eye, as to say, oh, that's just impossible. No. No, that's not the way to look at things. Just say, well, show me the evidence, which I have. Now let's discuss it, which has not happened. So as you're going to see, this is going to be a very long discussion of most about it is the discoveries I've made from mud fossils, which relate to biology that they didn't understand. This biology. Nobody knew about the interstitium. I wrote a paper about it in 2015, and all of this new stuff is based on that paper. If you read the paper and then read what they wrote, it's virtually identical. So this needs to be taken into account. I have all the research on it now about the enzymes and the bacteria and the fluid-filled highway, and how the ribosomes are able to get through the blood-brain barrier, and how the enzymes open up, and how the half-life of the isotopes are the way that biology works. It happens like this because of catalysts and enzymes, and you can only have those because of bacteria. I am deep into this, and I want to be part of this discussion. So this is my epic work today. Okay, my friends, this is going to be exciting, for me at least. And what I'm going to do is drag you through the research again. I haven't seen this yet. I got sent to me by a friend, Robert Gartland, said, here, take a look at this. What do you think? And this is about fascia. Now, I went into about maybe a minute, and it's basically the same thing I am doing. And I'm going to, but it's presented a little better than I do, So we're going to watch this, and I'll tell you this channel and so forth, and he's going to be presenting what appears to be the newest research on fascia. Now, I think I have actually the newest research on fascia, to be perfect. And I'm not kidding you. I'm just that. It ain't bragging if it's true. And I also have the Spurlock, which I got some here that are just going to knock your socks off. Those are the attachments of the fascia between organs and so forth. The organs are isolated in a bag of fascia all on their own, which has a certain chemistry that relates to that specific type of organ. If it's caustic, it's got to have some kind of chemistry and enzymes that work with that caustic environment. If it's, you understand what I'm saying. The stomach is obviously going to be different than your brain or your nerve tissues, that type of thing. So every one of those is a membrane. So we're going to get into, and the fascia is that entire membrane. It's one membrane if it does the whole body. And I wrote a paper on that in 2015. I know I keep saying it over and over and over, but I did. And it relates to every little bit of this, which is a fluid-filled network of tubing, really, that goes, and he's going to show it. He's doing a nice job. So I'm going to let him present it. Okay, here we go. You see, this just popped up in the last 10 seconds. Completely different discovery of strange 514 million year old slug rewrites our understanding of sea creatures. Everything rewrites everything every day. 514 million years ago, they're talking about the Triassic. And the Triassic rewrites everything. It was not 514 million years ago either. I know almost exactly how long ago it was, and it was extremely recent. If you read Velikovsky, you would understand, because he went to everywhere in the world and got the ancient sources that spoke of this, and they all had the same story, basically at the same time. A little variation here, a variation there, obviously. It's 3,500 years ago, and that's how long it was, 3,700, I believe it's approximately. And that's because it's all, everything I have is on the surface of the earth, and it's all preserved because of fascia. And that's why I wrote the paper, Fascia Facilitated Fossilization. Now, let's see what he has to say about fascia, because I just watched, well, I watched up to here, I guess. So I didn't get much of it, but what I did, it looked pretty darn good. So let's take a look. All right, so here we go. This is the Fascia Guide channel. And again, Fair Use Act. I'm not trying to steal anything from anybody, but I'm going to comment on what they are saying about the fascia. Now, here we go. It's the entire body.
Mankind has always been fascinated by how the body is connected. We have studied the skeleton, listened to the beating heart, tested the nervous system, measured muscle strength, and divided up the body piece by piece. Every organ has been dissected and analyzed. We've made great progress on so many fronts, but some of the greatest mysteries remain. The fascia, for instance, previously just seen as an insignificant membrane surrounding our muscles, organs, and skeleton, something we used to disregard and just throw away at autopsies. It's now one of the most exciting fields of science. Just another piece of the puzzle to help us see the human body as a whole. Today we know that the fascia is a network without a beginning or an end, and that it works in parallel with the nervous system and the bloodstream.
Okay, we're going to get into this deep, because the fascia is basically your immune system, and it is the delivery of all, basically all of the goods and services that your body uses. Fascia is the fluid-filled highway, and that's in the lymph, has all this bacteria and enzymes and ribosomes living it in it, and it surrounds your body, every organ, and if something tries to invade it, it comes over and kills it. If that organ is like a intestine, and the food is coming down that intestine, and the little villi that are in the intestine, it goes all through these little tubular looking things in there. You need enzymes to break those things down, and so that's where the enzymes kick in to break down all the food. All right, but it's in that same membrane. There's a membrane on every single part of your body called the interstitium, which is the fluid-filled membrane, which is what he was just showing you, all those little tubes. All right, this goes back close to 10 years ago, and this was fascia facilitated fossilization by me. And I also had fascia and salvomorphism, because I was relating it to fossilization. But I also said in there, fascia does the same thing when you're dead as it does when you were alive. Let's take a look at this paper. All right, I put a whole batch of papers up here. I put like 45 papers up here long ago. Nobody ever read them, so I just stopped. But I wanted to have them at least locked in somewhere. Now, this is fascia facilitated fossilization. Now, this goes to 2015, and I talk about the fascia, and I say if it wasn't for the fascia, it'd just be a gushy bag of organic molecules and metals, and the things that would dissolve in the rain just fall apart. Fascia is the reason you stay together. I called it the strings and springs in connective tissue. Fascia is only recognized as important in the last few years. This was, like I say, this is 2015. It was only, I started with fascia about 2010 or so with a guy called Gil Headley, and he was, I saw his video called the Fuzz speech, and he goes all over the world teaching about autopsy stuff. Anyway, so I got a hold of him and we worked this out about the fascia and the fluid-filled highway and the interstitium. So anyway, at this point, nobody was looking into it. I say fascia is only recognized in the last few years. Germany is the leader here, and they were. They talked to me, we talked. Anyway, when confronted with spectacular preservation in mud, I'm always asked, because this is the mud fossils, how is this possible? When I try to explain, I feel like a doctor with a patient, and his eyes are just glazed over. They don't even think, they listen. So it's hard to present this to somebody who won't listen. So unless you are a medical person, you may never even have heard of fascia. All right, so what is fascia? So then I go on to say what fascia is. Now, fascia is a coating on every fiber of your entire cellular structure, every single fiber in your body. It is the web of your life. In ancient times, it was called tunica, which means like a cloak. All right, the fascia has a job to do. Now listen to this. The fascia has a job to do. It separates everything. It protects and separates tissue and does it well during your lifetime. Guess what? If you die and are buried quickly and stay wet and compacted in fine, wet mud, which is the red bed, the fascia does not even know you're dead. And it keeps protecting the contents. I'm not kidding you. When they died and everything boiled off and they were inside that flesh, they're still in the same condition they were before. It's just that they're not getting fed. So they stabilize. Because they were boiled off, and there's a whole bunch of reasons that they stabilize, but the fascia was the reason that they all came out separate. And I mean, when I say separate, I'm talking separate. That's a lung. And this guy died almost absolutely as a product of being boiled, and he must have been like this in the flood, and the back is flat as can be. And it looks to me like this is exactly where a heart would be in a human being. All right, that's a left human lung, and it is human. It's been DNA tested, CAT scan, and everything. The CAT scan doesn't do much with these because they're transitioned, but the DNA test came back human DNA. So this is what we're dealing with, and this is all fascia here. And fascia coats the bone. That bone, anything that's collagen and keratins, they're the rubbery fabric bags that hold everything. They're the fabrics and fibers and gluey stuff. Now, the thing is, this has to be a certain chemistry because it's only dealing with blood, basically. So when the fascia here, the coats it just like in one little rubber bag, and then it has an exit here where it attaches to a next body part, in this case the wall of the cavity of the, inside of your body. Somewhere it's latching in to something. That's what these little bits do here, these little triangular pieces. And then there's one at the top that holds it too. So that keeps this thing from rolling around. But it also has to transfer all the fluids from here to the next organ, or the next wall of the cavity of your abdomen, or wherever it's going. Muscles, and I can show you muscles, and I have one right here. Right there, that's the flap I'm talking about, and I call that the Spurlock. And I have one here, it is spectacular. Spectacular. That's in bold, highlighted, big letters, spectacular. All right, you see this right here? This little latch right here, this triangular latch, right to back to about here is a really heavy duty lock that locks this piece into the next muscle group or into your body, or in the case of a lung like this, it's the same latch, the same little triangular latch, and it latches it into the cavity of the body so it doesn't go floating around. Now this is muscle, and that apparently locked it into another muscle, and I have another anatomical shot of this being ripped away. Mine obviously is a rock, this one here, but this is actual body tissue. Now, what are we looking at? We're looking at that right there. That's the same latch. So this down here was whatever it was. In this case, is muscle. And this is latching this little triangle up to make connection to something else. This looks to me like it's a tendon or a muscle attachment or something. Because wherever something attaches to something else, it'll rip out. It has to have a heavy-duty attachment. And these suckers are heavy-duty. And you can see back here, there's a really scruffy spot, and from there coming forward is the triangle. And right at the tip of the triangle, there's two little dots, and that's where the fluids transfer. And I think they must be, I'm not sure where they are here. Maybe there's three, I don't know, maybe. But this is ripped here. That's why they're showing this. This is an injury. But it's the same as this right here. And every part of your body has one of those. They're everywhere in your body. And the fluids have to go from each part of your body, from your biceps and your everything, to the next part of your body. It's just the way it works. Because it's a fluid-filled highway, so it has to move from here to there. Now, is it the same fluids here as it is in the next organ? I don't think so, because they have totally different chemistry. All right, we're going to be talking about these, what I call Spurlocks. Everywhere where an anchor, where a muscle or a bone or whatever tendon anchors, or a muscle attaches to another group of muscles, you're going to have one of these locks. It's going to be a really heavy-duty jobber. Now, a lot of them come out of the bones, a lot of them seam together. Primarily, I find them in the lungs. That's where I see them easily, but this is just this piece of meat, so they're everywhere, but they're everywhere in your body. And I mean, there's just literally thousands of them. You know, look at this. Everywhere one of these bunches of muscle meets together, you're going to have a latch. It's going to end up locking somewhere. All right, just some of them lay flat like this, but the tendons all have little anchors at the end. This is an abrupt transition from muscle into tendon, and the tendons lock into the bones or wherever they end up going. This is what they call a ponerosis tendon, the flat tendon. And then the rope tendons, the ropey ones, are just a regular tendon. And these are ponerosis. All right, so you know about the little flap here. That's this little flap right here. Now this white heavy-duty stuff, this is the heavy-duty part right here that flanges away from the white anchor. You see it? And it's pretty heavy-duty right at the bottom. And then it still stays pretty heavy-duty for a ways up, just like this is. And then it sort of tapers off, and you get into the muscle, and you get an abrupt transition. Now, but underneath this, underneath there, we can see the muscle just like we're looking at this muscle here. But that's the flap that would have attached to something else held here and looked like it would have held right into there, and this went right into the webbing of the other fascia that was on top of whatever that other piece of meat was, or I'm sure this was locked into another muscle. Well, okay, my friends, this is just over the top crazy. Remember this triangle, which is the flap that locks into the next organ. Now, If you can see this round thing right here, just take your time. All right, take your time to focus at it. All right, you see that bump area here, right here? These all have little tabs where they lock in. This is the bump that locks it in to the next place where it's going to lock into, and it has like a stub into there that locks it in, and the membrane sort of separates and changes. Normally a membrane has 2 layers, and that's it. Everywhere it goes, it's like this. When it hits this point, they sort of interchange like this, and I will show you that because I have one. It's right in that process, and it's spectacular. Now, if you can see this spot right here, you see we got a round spot here, and then we have this spot up here, which is the really gnarly attachment. This thing here is just tough, tough, tough. And that's another little round lock in. I call it the Spurlock. So it's locked in here, and it's locked in here. So this is locked, and this locks into the next piece. And the next piece, I'm not sure how it does attach, to be honest with you, but it locks into the next piece somehow with this flap. And it's just, well, I suppose you could look it up in a human lung and see how it latches, because this is the same latch. It's the same on all of them, and it's the same on the human body, between organs or whatnot. Let me show you something. Alright, this is a little graphic, but this is just somebody butchered meat. And these are the way the tendons invest. This is the rope style, and then this is not really a ponerosis, but it's in that area. And they have fibers that run off, and they go to other attachments and pull this way and that way, and so you can do all these different things. Very, very sophisticated system, very sophisticated. Now, these, I think that might even be a Spurlock right there. You see that little round spot? They latch in with these round spots, and they're all over the body, these round spots. That's what I found. Okay, I got the microscope cooking. We're going to be looking at the fascia, which we've been talking about, which is, this is a lung, which has, and it's a lung right here that's in the microscope. And this is pleural, which is really just a very heavy coating of fascia. Let's think of it that way. Now, this particular lung is also, it's all the way down to the alveoli. You see these, all these little round spots here? Those little round things, you see them? And then you see this little red dots all over here. All that, this is all alveoli in the lung. This is where air went in, and it transpired into this red blood so that you can keep your oxygen up. Now you see this spot right here? That's the Spurlock from that lung. Just like it's the one on this lung. This one's all eroded away, but there's still that heavy duty jobber is still down there. That's what's locked into the actual lung. All right, and I believe right there is where that anchor spot is. You see how it's like circular here? It's a little bit different than what's around it, and then you got that flap. Well, this one you really, well, you actually can see a little bit of it. I would say that's it right there. But this is much more built up. Now, I have something that's going to just, it freaked me out when I saw it. I just couldn't believe it. Don't forget, this is a membrane. Now, let me show you what a membrane looks like. Alright, that's what a membrane is. These are phosphorus, phospholipids, which means there's 2 membranes, which are phosphorus, basically, phosphates. And in between is the fluid part, which is hydrophilic and hydrophobic little things in here that keep this separated apart. And the only way in and out of here is through these little ion channels. So they're extremely regulated. Your membrane is what protects you. It's not going to let anybody just come in. Now, if any of these here are not secured very tightly, you're open to invasion. These are called tight junctions. And then a little down further, somewhere in between this here, there's adherons, and then there's desmona junctions. There's 3 layers of junctions to keep this sealed. But you need the right enzymes and the bacteria to keep them in good shape. So that's why people get sick, basically, invasion through this. That's basically what most cancers are. And every single bacteria has an enzyme, and that enzyme has a job to do in this membrane. And it's in that fluid-filled highway. So it's extremely important. And here's the thing. This is just new. This is the Interstitium of Newfound Organ. In 2018, they wrote a paper on it. Same paper that I wrote, basically, almost identical. So I got a feeling somebody's keeping an eye on my research, because I didn't get it from them. I discovered this myself. And at the time I wrote my paper, it was fuzz. It was called fuzz. All right, this is only like a minute or 2 read. I'm just going to go right through it here. So I told you the fascia has a job to do. It protects your tissues and does a good job when you're alive. And it does the same thing if you die and are buried quickly. You stay wet and compacted in fine mud. The fascia doesn't even know you're dead. It keeps protecting. This is the cause of mud fossils. This is what protected them, and they've eventually became what's called nucleophilic invasion and substitution. Now, the mud fossils are exact copies of original creatures separated on these fascia and skin separation planes. Wherever there's a collagens, the fascia separates every cell and structure from its neighbors in life and death. Fascia protects every bit of you from invasion. It's your immune system. It protects every bit of you from invasion, including a complete fabric bag that is your skin. That's your protective layer. Your skin is not just one fabric either. You have tough skin, fine skin, all kinds of thin skin. All fascia is made of collagens and keragens, the fabric creating fiber and elastins in a sheet that wraps everything individually and then comes together as one giant system. It's all just one huge system. It's one fabric-filled highway. There's over 20 types of each collagen and keratin, and some protect from acids, other from caustics, other mineralized for strength. The variety is amazing. These fibers are infused with silicon. Silicon seems to be important, and I really don't understand it all that well, I got to be honest with you. I think Marguerite understands it better than me, so I work with her on some other issues about bacteria. Now, tissues in your body are varied and many, all kinds of different tissues. Some vascular tubing completely impenetrable, and connective tissues are highly mineralized. Skin has 50 times the silicon as other tissue. Silicon carbide is the fossil version of skin. See, here's the key. I didn't realize, I was looking at the skin and seeing there was 50 times the silicon. Well, that's because it was in extremely silicon heavy-duty waters. All right, I'm talking about the fossil version of skin. It has a 9 on the hardness scale, which means hard and grippy. That's the silicon skin, and that's what they found in the paper that Yale wrote. Here's the paper Yale wrote. Hold on. Well, I might as well show it to you. Why not? It was all silicon, and it was one worldwide flood covered the whole world. Exceptional preservation, soft-bodied creatures promoted by silica-rich oceans. That's the 50 times the silicon. Now, the reason was because we almost got hit by Venus, and it virtually caused a worldwide flood. That's because this is a worldwide. The whole world. Worldwide. Worldwide. All right, and they claimed this, it was before there was even supposed to be any life, and these are all soft tissues, which is not correct. So Yale sort of distorted this thing, and I gave all my work to Derek Briggs, so this seems to be a distortion of what I presented. But they did come very close. They admitted that there was creatures here they can't possibly account for, bizarre, and then they were perfectly preserved, soft body creatures. Exactly what I wrote in 2015. So again, come back to here. Let's finish up this here now. And then I come down here. I say that it has 50 times the silicon in the mud fossils. Mud fossils contain all the body parts, and each is protected. Fascia fossilization. Fascia is a part of the connective tissue and invests itself in bundles at certain places in the body. And then guess what? The investment sites correspond to ancient acupuncture points quite closely to where you attach one organ to the next, and they put needles in there. I don't know. I mean, they put needles all over your body. But anyway, so the acupuncture points seem to be in some of these areas where there's a lot of connective tissue. Now, it is now suspected that fascia connective tissue may be fiber optic. That was my suspect. I don't know. It could have. I don't know fiber optic, but it communicates with every cell in your body. So it's a good chance that it is. And anyway, fascia was thought to be mere packing material, virtually inert, long been ignored as a subject of research. They just throw it away. They cut it out and they throw it away. When I first talked to the guy, he says, "Yeah, well, that's fuzz. You don't even need it." I said, "You don't need it?" He says, "Yeah, we cut through it. You take a chunk out, throw it away, get it out of the way." I said, "Yikes." And that was in about 2010. Now, so it was just thought to be packing material. So recently, however, there had been new equipment that allows the viewing of the fascia. Now, this is in 2015. They had just released this, and you could see that there was something going on with this fluid-filled highway, which I knew was in there. So the fascia is a tubular fluid-filled fabric network. Fluid-filled highway. Tubular fluid-filled fabric network. Instantly reconfigures itself if damaged or pulled, and because you can know all that stuff all the time. It appears as if a fiber optic wired network is redirecting the broken fibers. That was just silly speculation, to be honest. I believe fascia is the fiber optic style network in creatures as it connects to every cell and is complete system. It does go through all the lymph, and the lymph is the tiniest little fibers you ever saw. I feel this is logically the wiring for send and receive communications. It appears fascia may be much more than simple fuzz, as doctors often refer to it. I talked with this guy, Werner Klinger, at the University of Ulm, has done some excellent work on fascia, and we were talking about water having memory. So you know where water comes from because it's in the memory of the water, in the molecules. And I also feel quantum numbers, possibly from folded proteins, are the source of the program code. Well, we know there's program codes in the DNA. Quantum numbers may give you exponential expansion of the numbers, and plus, in the DNA codes, the amino acids, there's like 20 of them. So instead of having a 1 and a 0 for programming, you got 20 codes for each one of the things in the bits. The programming in DNA that's stunning, and 98% of it is not junk DNA. That is there for a purpose now, and I believe it's got related to memory and emotions and all that stuff. Now, here we go. So I'm saying all of this stuff is related, but that was all pure speculation. I admitted this is just speculation on my part, so don't take it serious. That part. Now, as a component of support and posture, fascia also suspected to be a source of many pains. Yes, 100% for certain. This is the approach that science is taking to fascia. Medical science is interested in life, not fossils. How does the system work in the life process? How to detect and repair this as a system? I've worked on that too, and I understand how to actually get your fascia moving again in your body, because there's nothing more than a system of glued up little fibers at some point, and you have to get them moving again. All right, so my approach to studying fascia is somewhat different. I analyze the fossil fascia. I look at the chemistry. What can be explored? How are these rocks formed? What causes the elements, the metals and such? Because they concentrate metals here and there. What are the steps in sublimation and solvomorphism? That was the other paper I had. What is the process that creates fossil fuels? because they are from fossils, the blood primarily. And it results in organic compounds and volatiles. How does pH affect the process of fossilization? What effect does wet soil, which is spontaneous electrical potentials, have on organic matter? What is the molecular structure of different bio areas in the body? You can have all kinds of different molecules and different chemistry depending upon where it is in the body. Like connective tissue versus lungs. How does the chemistry react during death? What is the process of decomposition and purging of fluids? What were the conditions of burial? What forms of chemical invasion are at play? How does his tissues form layers, and how do they separate and transition? What is the process creating voids and vugs? How does metal seeding happen in crystals? The process leading to crystal formation of percomorphism, also called solvomorphism. All these little bits add up to, in some cases, exquisite, exact anatomical stone replacement body parts with every structure and even the colors of the original flesh intact. And then I go down to some stuff, and I had to take all this down because I got destroyed. So forget about everything after that. But up to there, that's just what it is. And like I said, the original colors just look like a piece of flesh. You could put that up a plate next to a piece of meat, which I did, and you almost couldn't tell the difference. You almost couldn't tell the difference. Only this looks more marbled, looking like more delicious. I mean, come on. All right, let's be sure you understand exactly what I'm saying to you. This is the securing place. There is a place back here where it locks right in, right there. All right, that locks this piece into this chunk of meat, wherever it was going to, it's muscle of some sort, apparently. Now, from here, that flap comes up and lays on something else and locks into it, and I think with another one of these little securing tabs. And I think that's it right here. You see it? I can't be sure of that, but I am pretty sure, because wherever they lock in, they have one of these little tabs, as far as I can tell. So that's the piece. Now, I'm going to show you something on the lung. The lung has the same little tab down here at the bottom. Okay? So now, what would happen if this piece here didn't stay with this? It left and stayed with the other piece, which does happen. You're going to leave a chunk missing right here where that would have been. And where would that have been? It would have been right at the corner of the lung. Okay, so if this ripped away, you're going to have a chunk laying down inside there. And guess what? I have one that eroded in such a condition that it ended up losing that piece right there. That's the piece that would have locked it in. All right, it stayed with the other piece, or it just fell off, whatever happened there. Now, I got to show you something that's going to freak you out. It freaked me out. It still is freaking me out. I got to be honest with you. This right here. Now, don't forget I talked about membranes. Let me go make sure you understand the membrane. All right, believe it or not, this is a membrane. That's this membrane, right? Just like that. Now, remember I talked about phospholipids? Phosphorus, when it boils down into aluminum silicates, which is what you got here, aluminum silicates. And that's one layer, which is from the phosphorus is the top layer, and from the phosphorus is the bottom layer. And in between is the fluid-filled highway, basically. Well, it's fluid. It's all fluids. It's this layer right here. Now, there's some things that are going to be happening up in here. Now, look at this. You see that bump right there? I don't know if you're going to see that. That's the bump right there. You see it mounted up right there? That's the bump here that locked this which was nothing more than this piece, right, just like this. Hold on, can you see that? Well, let me hold it in a different way. Let's say this piece right here was attached to this. It was that tip, and it broke off, because this would have obviously been much bigger. But the whole thing broke off, just like it would have broken off right there. And this is this tip. That's the bump that latches it in. Now, you see how it's nice membrane? This is how it's supposed to be, like this. It's supposed to be like that continuously. You're not supposed to have this. You see this? This is where it latches in to the next organ. And it latches in like this, and it appears to have filtered in and broken right off. And this piece is all on its own. And when you put a little tiny touch of water on there, you see how red that is there? That's where that locked down. This piece here locked straight down. And here's where it started to separate the membrane and change to go into the next membrane. It's supposed to be like this, all the way down. All right, but it's not. When you hit this area, and look here, this is where some of this fluids are being transitioned over, it looks to me. And over in this area, this looks like a separate piece coming off here. In other words, it might have had two attachments. One attachment coming this way, one coming this way, but this one broke right off. I think that's what's going on here. I think that attachment was coming out. I looked at this, and it appears to be a secondary attachment coming right out of that little spot right there, heading out this way. And that happens all the time. You have one coming here and locks in, you have another one coming over there that locks in. It happens all the time. You see this? Every one of these spots is an anchor. There's literally probably thousands and thousands of these in your body. Now, they're very tiny for the most part, but this is from a gigantic creature. And again, that is going to be the membrane. So all of these are membranes. Everything here has a membrane on it, and depth, like layers after layer of membrane. Hold on, I got other shots showing how many depth layers there are. Look at this. All of that's covered by a membrane, and it separates that from this, which is all covered by a membrane. It separates that from this, which they're all covered by membranes. From this, from that, all covered by membranes. That from this, same thing. All the way down until you get down to the bones of your back. They're all separated by their own separate membranes. Membranes are just everywhere. They're just everywhere. And there's some places where there's 3 or 4 attachments at the same place. So I think that's what's happening here. You got one really solid attachment, and then you got another one that was going to be running off this way. I don't know what it would have done. And then that locked in to, like, say, the shoulder here, up here, and this came down to run down to here, or something like that. And it just snapped right off as the attachment spot. And when you look at this, you can see how it just like laid flat and attached. I have grip skin that is just like that. Now, you see this here? That's the bottom of the button. You see, that's the bottom of the button. If we look at this correctly in the microscope under the right light and the shading and all that stuff with a little moisture, you'll see exactly what that is, and it is just saturated with blood. And then we have to look at these to see what they are. I haven't really given this a real good look at yet, so we're going to do this together, as will be my policy from now on. Right, this is the attachment spot right there, I believe. Now, it's hard to make it out, but you see all this red blood around there? I'm going to put a little moisture on there, and it usually changes things pretty quick. Now, it's going to be very reflective for a second, but I'll dry it up, and we'll be able to see, hopefully, a better way. You know, we can see the attachment spot a little more clear. Whenever you see blood, normally there's some kind of attachment going on there. And that's a lot of blood. And I believe that right there is the attachment spot, per se. And this would have just laid right into the next piece up here. And I believe this might have even been where the fluids left this piece into the next piece. Now let me come up with the light a little bit. You have to play around with this a lot. And a lot of times you can see something new just by changing the light. And focus down a little, up, down a little, down, a little up. See, this down here, I think, is right where it actually engaged with the next piece. And this was part of the latch that locked it in. A lot of blood right in this area. Right over here, a lot of blood. Now I'm going to back away down where this would have just been laying on top of the basement layer. You see, we lost all the blood, not lost all of it, but a lot of it. And we still got this aluminum. I'm almost sure this is aluminum. We got blood here and there, but mostly we're getting into a gritty area. It's coming back to where there's some grit. And this is where you get the gnarly stuff. And back here, I believe it's somewhere around here. Yeah, I think that's it right there. I think that is the back spot right there, where it locked in. Don't forget, there's one at the tip, and there's going to be one back in the back. And they always appear to be round like this. And there's usually some pretty good blood around it, but there's normally a pretty, obvious border. And this appears to be the exact dead center. Now I'm going to continue to go back further here. And this is where it's going to get a little gnarly. Because that goes right after the spot, and then here's where it all fractures up in the back. Now, that is not a normal membrane at this point. That shouldn't be all black and white like that together. So we're looking at the side now. Let me focus in here. You see that? This is where it weaves into the next body part. So let's go sideways again. We're at the top where we have all of our aluminum. Alright, and then this is a sideways version right here. Now this is the bottom part where we have all of our aluminum. But right here is where the two meet together and they fuse into each other. Let me see if I can get it. I'm going to have to get a real steady shot of this. You see, we lost it. Somehow the tubes are bringing those fluids in. And this is where everything attaches together and locks solid. But the fluids have to be brought in, and I think I know where that's happening too. Let me find that spot. Hold on. Here it is right there, actually. There's the black and the red. Hold on, let me put this in the holder. Alright, I just put a little water on this spot, on the side. Now, anybody that knows about the mud fossils knows about the black and the red. The black is primarily vein blood, and the red is primarily artery blood. Sometimes they mix around a little bit together. Now, what we are seeing here is some kind of transfer of fluids. And it looks like there's red blood up in this area, and this looks like black or vein blood. Now, this is just one side of this thing, but this area right here is, I believe, where it starts to feed right into the other piece that it's mating up with. This whole area right in here. Totally different than a normal part of the membrane, just way different. Now hold on, I'm going to turn this around a little bit. Alright, keep your eye on this. This is the other side of the membrane where it meets up at the very end. So we're on the complete other side. Yeah, it was flipped over on the other side we looked before. Now we're going to come down this and look at it as we go. You see? Oops. Every now and then you're going to see some red blood coming in. There's going to be arteries here and there. Alright, this is some heavy duty, whatever it is. You know, that's part of the membrane. That's the same as like the membrane up here, but it's strappy looking. I don't normally see them looking that strappy. But when I show you the top of the membrane, you'll see it's the same as this when I get into the aluminum up there. Now, let's just keep coming down the best we can here. Oops. It's hard to focus this thing here. See that? There's some crazy looking stuff that happens in these membranes as they transition. You know, I'm going to have to freehand this. Alright, this is going to get a little shaky, but I think it's going to be better. I just can't get close. I can't work it with the holder. No, here. So here we were at the very end. Alright, you see all the red? That's where it invested right at the very end and changed over these different layers. And coming back, it turns into the normal, you know, membrane. And it just keeps going back. But you got a little red blood here and there. Now, I'm going to show you the top and the bottom, the difference between the two. One of them, the top is all fibers, and the bottom is all sort of flattish. So let's prepare for that. But you see what I just showed you here. There's the membrane. You go up as it tapers up to the end. Right at the very end, it comes together and makes this situation at the very tip, which ties it into the next organ or the next body part, whatever it is. And that's where all the red blood is, right there. Alright, so let's take a look at the top and the bottom. Alright, this is an absolute, total, complete mind blower. These are the fibers that you see in the fascia. Remember those fluid-filled tubular highways? Well, that's them. These are all that stuff. Remember all that little fluid-filled fascia, little tubes, and he was pulling at it? That's what he was pulling at. This is so identical, I can't even believe it myself. This is the fascia. Now you see the red stuff in there? That's blood. And you see all the other fibers? These are all the fibers in fascia. And these are some of the blood. Look at this. Look at this. There is no difference, none whatsoever. Only these fibers have been turned into aluminum silicates. Some of the red blood is still in there. This is just phenomenal. I mean, I can't believe it. Every time I look at something, I find something new. It's just beyond my belief at this point. But you can see that is the fluid-filled highway of this tubular network. Unbelievable. Now see, this side was just laying down on something. There's no, that fluid-filled highway is not here. You see it? The fluid-filled highway is the top, the fascia. This is the basement layer. All right, you still got a lot of blood and stuff like that, but it's a flat mat, basically. Isn't that something? All right, so I'm saying this whole big chunk is a Spurlock. All right, remember I said coming down there was going to be a little piece that broke off on the side, and I can guarantee you that is a fact. This is the piece that broke off. That's the blood supply to it, and this is the fibers that were leading into it, and it just snapped off there. There's no other spot like that on here. Now don't forget, I say that is the spot where the thing was going to come off like this to go to another place, and it would also have to send some fluids and everything else with it. The interstitium would have to go with that too. So where is this on our sample? It's right over here. Let me turn the lights on, and you should see it right here. Now remember, this is the spot I'm talking about. We're just looking at. This is all of the fluid-filled highway up here. These are all those little fibery-looking things, and this is the piece that would have run off that way. Other than that, this was the Spurlock. This is just another piece hanging off the side that we were looking at right there. That's going to go to another connection. You see it? Other than that, this is what a Spurlock is right there. That's what the Spurlock is. And that's the main attachment right there. And these are all of the interstitium, which is at the top, and we know this. It's this stuff here, right here. All right, and at the bottom, here's a real red, bloody spot that anchors into the newly fused interstitium. Back here it's just totally membrane, just normal membrane, but then when you get up to here, it breaks off into another piece, and here it breaks off into attachment to another piece. All right, and back here it's very gnarly. This is quite gnarly back here. This is what not normal. You see these really spiky looking things? That really spiky looking thing right there. And I have them in all my samples have the same gnarly, really nasty looking spot that locks in. So I hope that makes sense to you. I think it does to me. And there's so much evidence that I can support what I say that it's hard to dismiss it. And the creatures were astronomically large. I've shown this so many times, it's just virtually undeniable at this point. So, will we continue to deny reality, or will we move forward and see if it has any significance? I think it has a lot of significance. It does to me. But it doesn't seem to have a lot of significance to a lot of people. And that's hard for me to understand. Anyway, that is the Spurlock. And I got some, I can show you dozens of them, literally dozens of them. That's a Spurlock on this lung. That lung died flat in the flood again. And this one has half of the pleura detached, and the rest of the lung is still perfect. Flat as a pancake. There's the Spurlock. Look at this one. This is another one. That's a lung. You say, oh, no, Roger. Yes, it is. And there's the Spurlock right there. You see it? That's the Spurlock. And that's a lung. But when this one died, it was flat like this. It depends. See, that's the flat spot. They all have a flat spot. Well, not all of them have an exact flat spot, but most of them do. And they boil out of the bodies. The creature's bodies boiled and made what they call a red bed. It was just nothing but flesh. And then the body parts, the organs and stuff, started landing on there, and they're just laying flat, making flat spots. And here's another one. That's the Spurlock on that little tiny lung. And here's the two little tubes coming down, I believe, that are feeding it with the interstitial fluids. And this is completely eroded away like this other lung up here, wherever I showed you that. This is like this one. And there's its flap right there. It's just like this flap right here. But this is from, like, I don't know, a monkey or something. I have no idea what these things are. So I do know 3 of them are human DNA, and I had them tested. And they are human, and 2 of them are huge, giants. And I mean big, big, big giants. And one of them, this one here, is our size. That I had tested too. And it was a very, very good test. It was expensive. It took months to do. It was a PCR test, 3 samples, tested positive for a couple of different regions in the mitochondrial DNA, which means it was a human, modern human actually, the same as everybody else's mitochondrial DNA. Because that's the same on everybody's side. Is this a couple of genes in the mitochondrial DNA. Everybody has the same ones. When you get into the other side of the DNA, the father's side, you get a lot of variation going on. I believe that's how it works. But the mother's side, the mitochondrial DNA, there's some things that we have. Everybody has the same one. You know. And these had them, so they were human. Okay, so we saw the fascia, and I'm telling you, I believe that's the fascia right above the membrane, exactly where it should be. Now we're going to go back to the video, and again, this is Fair User Act, and not trying to steal anything. And what we're going to do, let me turn that off. Now, what we're going to do is watch his video and listen to what he has to say, and it does appear to be well done. So here goes. Again, I only got up to a minute or 2 in here.
Mankind has always been fascinated by how the body is connected. We have studied the skeleton, listened to the beating heart, tested the nervous system, measured muscle strength, and divided up the body piece by piece. Every organ has been dissected and analyzed.
Well, wait a minute. Every organ maybe, but I'm going to tell you right now, the major organ in the entire body is the interstitium, which the paper I wrote about that system, that fluid-filled highway, is brand new. That's in 2018, and I know, I'm almost absolutely certain they don't know about the Spurlock, which is this little. It's a special little piece. It's a little organ. That's different than the rest of the stuff. And every one of these investments has to be able to transfer their fluids into the next one. And they were acupuncture points. This could be very important. So here we go.
Analyzed. We've made great progress on so many fronts, but some of the greatest mysteries remain. The fascia, for instance, previously just seen as an insignificant membrane surrounding our muscles, organs, and skeleton, something we used to disregard and just throw away at autopsies, It's now one of the most exciting fields of science. Just another piece of the puzzle to help us see the human body as a whole. Today we know that the fascia is a network without a beginning or an end, and that it works in parallel with the nervous system and the bloodstream.
Are you struggling in
Okay.
calculus? Having a hard time understanding your professor?
Yep.
Do you wish there was a better way?
Absolutely.
Emma experiences very same challenges.
And I've always been really awful.
But let's begin 500 years ago in Renaissance Italy.
Den moderna förståelsen av människokroppen
The
grundas
modern understanding of the human body is based on the fact that dissection used to be prohibited and taboo.
vad man gör.
So some enthusiasts dug up dead bodies and carried out autopsies to understand what we really look like. They did very detailed drawings. Leonardo da Vinci was involved in doing these kinds of drawings.
Det var ju Leonardo da Vinci. Redan där börjar ju det här.
And that was when we started to see the body as something you should divide up, in different parts and organs, clearly distinguished from one another. And eventually that leads to, let's study liver function, let's study how the lung work. And that generates a massive amount of wonderfully detailed knowledge. But this sort of knowledge, this type of mentality, assumes a fairly weak foundation for a holistic understanding. Because then you need to take a different approach. And we're not so good at that.
All right, when he's talking about holistic, that's what I'm talking about. You have to keep your membranes in good shape and keep your bacteria in good shape so your enzymes are producing the products, the ribosomes, you know, and the enzymes and the proteins to build your body and to use the other things to clean it up and do all that stuff. That's all done by bacteria. That's the holistic approach. They're looking at each thing as a separate little device, and it's not that way. They're not separate devices. They are contributors to a holistic system. And if this doesn't work, something else is going to go wrong over here. Everything's going to go wrong somewhere. You have to have these enzymes to break down these products so fast in these, what they call half-lives, to do biology. It just cannot happen without the enzymes and these catalysts doing these breakdowns millions of times in a second. It wouldn't happen for years without these. This is not a game. This is real. These enzymes have to be produced by bacteria. So you kill your bacteria, you're in trouble. And a lot of people have a lot of bad bacteria, and we have no database, and there's no way for you to get a quick exam of, is my bacteria okay? I don't know. Who knows? Nobody knows. All right, so he's saying we don't take it holistic into account correctly.
It's not vitally ingrained in our tradition and culture to think in that way.
Industrialization, from the late 18th century up until the middle of the 20th century, pushed us to see the details rather than the big picture. It fueled our belief that everything can be substituted, everything can be replaced by a new worker, a new hammer, and eventually a new body part. The idea of human spare parts was forming. In schools, we started dividing everything into subjects. We all needed to know the same things and to learn them by heart, but without much thought for the deeper meaning behind them.
Hela vårt utbildningssystem från det vi är.
From a young age, as we grow up, our entire education system is built on dividing things up into different fields and subjects, starting at high school, if not sooner. I don't want to disrespect this method, as a detailed understanding is important. But if this generalist, almost philosophical approach is too dominant, if you believe that separation forms the basis for understanding, then it becomes very difficult. In the tradition of thinking we have become used to, holistic views seem somewhat intangible, which is problematic as the human body is interconnected. It's actually a whole.
Vilket är ett problem, för att kroppen hänger ju faktiskt ihop. Det är faktiskt en helhet.
So what happened to modern day mankind? We'd rather take a painkiller for a headache than prevent the pain. And why do we insist on eating unhealthily when we clearly all know better?
Well, I can tell you the whole story is money. It's all about money. And this glyphosate that I just did a video on, that I believe, well, I know it kills bacteria. And they spray it right onto the crops just before you eat the crops. They spray it on right at harvest time. And that stuff lasts over a year in the product. And it is saturated throughout the food chain now. And it kills bacteria. You eat that stuff, I would assume it's going to have some effect on the bacteria in your body. I can't prove it. But I would assume it's a herbicide. And your bacteria are herbs. little growing creatures inside of you, and if you kill them, and it's designed to kill bacteria, fungus, all kinds of stuff, and weeds. The only thing it doesn't kill is stuff that they went in and engineered the genome to say, oh, don't worry about that, you can eat as much of that stuff, it won't kill you. Well, that plant won't die because it's got some kind of new genetic engineering in there, which is but everything else dies. And then it eats all that stuff because it's just being harvested. And they know this. The studies have been done. They know. But they say, oh, that won't hurt you because you don't have this shikanite pathway, which is what it works on in plants. Well, I don't care what it works on in plants to kill them. We don't have it. Yes, that's correct. We don't have that pathway. But we eat those plants that could kill our bacteria, and that's our pathway. That's our good bacteria. Is that happening? I can't prove it one way or another. But I think studies should be done on that. The kids are getting autism now at a rate that's just staggeringly high. And I believe, from the research that's been done, it appears to me that it's the gut being slowly destroyed. And as kids especially, because they're just establishing their immune system at a very, very young age, and it's destroyed them. Whether it's from eating the glyphosate stuff, I don't know. Because it could be from anything. It could be, but a lot of it's from antibiotics and not replacing the probiotics. And I just have no idea why they won't go into this fecal matter transplants, because that's where all of that fecal matter is in your gut. And all it is just put a good bacteria in somebody else's gut. That's all it is. You put new bacteria in. That doesn't seem like such a big thing to me. And it does seem to work very, very efficiently. And there's very little side effects, they claim. This is what the claims are in the research. I give no medical advice whatsoever. I just report on the research. And fecal matter transplants, look it up on almost anything that has anything to do with a gut issue. If you haven't got, or let me put it this way, the studies they did were primarily for this Clostridium difficile, which is a bacteria that is very, very aggressive in the gut, and they almost can't get rid of it. But they do the fecal matter transplants. I believe they have 95% right away. People get better. So it's not like, and there's very little side effects from it. So it's not like it's not ever been used. They only have it authorized for that one use. Why? We need to know if people's bacteria levels are high or low. I would think that's to be the starting point. All right, and that's research. Research says let's do a big sample, a broad sample of everybody's fecal matter swabs. That's all. That's it. Simple as that. And then see what they all contain. And if people send in the symptoms they have or issues they have in their life, along with their sample, it goes into a database, and everybody like this, and one of the guys up here, and everybody up there says, yeah, they get up and they throw up every morning or whatever it is. You know, I got lung cancer. Down over here, they got pancreatic cancer. All these different cancers are linked to different organs. Different organs are linked to different chemistry. Different chemistries are linked, I would think, to different membranes. And the membranes are being penetrated. Why? Why is that specific membrane being penetrated and the pancreas is being damaged, but your lungs are okay? different membrane. What's the chemistry controlling that? The chemistry controlling that is a bacteria creating an enzyme, and if the bacteria is not there, the enzyme won't be there, so you don't have that chemistry for that specific pancreas enzyme that is eaten through the wall and through the membrane, so you're getting invaded. We need to have a bacterial database of just a nice clean database of everybody, giant groups of people. And then even you will find maybe that even like in Northern Ireland, they have a terrible problem with autism. Now, why? Is it just the population? Is it, why? I think they're a little higher than most. Now, if that's the case, is it related to the water? Is it related to the air? Is it related to the food? Is it related to the health care? Is it related to antibiotics being too abused? Is it related to drug use? I mean, what is it related to? You have to be able to get a database before you can even draw any conclusions. We don't have the database. There's no way we can proceed forward without the database. All right, I'm just going to run through a few lungs and the Spurlocks. That's clearly the Spurlock on this lung. Now, what kind of a creature it was, I don't know. Died in the Great Flood. That's the flat spot. And that is the Spurlock right there. So it was a lung, you know, they get all bent up and stuff when they, but the sac pretty much keeps them intact. This is a lung right here too. Now that's the Spurlock on this one. I don't know what that's from. Again, they're from all kind of crazy things, but that's where the plumbing through the side was. And that's the Spurlock right there. Now I'm going to show you something that's really going to freak you out. You see the different structure. Where do you see this? Okay, I showed you all the little Spurlocks. That is another Spurlock right there. This is on a tiny little lung. You see the little latch there? Same latches on all of them. You see it? And there's a gnarly spot right in the back here. See the round spot right there? Now, this is a lung. You see all these little holes? Hold on. All these little alveoli holes. Those are little alveoli holes where the lung is a lung. And again, it's got the same latch they all have as the Spurlock right there. On every body part has one. Now, I'm going to tell you something else about this particular lung. You see what's sticking off of there? Those are magnets. Upside down, sideways, doesn't matter. They stay on pretty solid. You know why? This is an iron lung. You see all the little cavities in there? Those are alveoli. You see the little white spots there? That is the pleura. You see, I can focus in on this. We'll actually be able to see some blood in here. And again, this, my friends, is an iron meteorite. It came through space and cooked and left some of the little white fibers. You see all those little white fibers here and there? Here, all here and there, those little tiny white fibers. This thing was absolutely 100% coated with them, just like my other lungs are, until it came through space and a lot of them cooked off. But some of them stayed. And you see the blood is still in those alveoli. You see the white fibers? Those are the same fibers that are in the lung I'm going to show you right now. All lungs have those same fibers. And here's what they look like. Right here. There's those fibers. They wrap around the lung. Now that little tiny lung came through space. It cooked up. Now, I don't know why it's in space. I'm not going to try to explain that. It came through space. It cooked up. You can't get these lungs on these things to turn into metal, iron, until they reach about 2,300, 2,500 degrees. And at that point, they start to turn into iron. But you can't get them up to that temperature on Earth here unless you put them in a furnace or something. And that was clearly a lung. It has all of the anatomical signatures and the Spurlock. So let's take another look at it. But this is the white fibers that I'm going to be showing you in that lung in the microscope. These are the exact same fibers, only all of these cooked off in that other lung. Not all of them, but most of them. And the other holes that we see popping, those exploded out because the blood was inside it. It must have been wet, and it just popped. Those are popped out. They didn't sink in. They exploded outwards. Okay, that's that little bitty lung. It's the iron lung that's in the microscope right now. And these are the alveoli. And I'm telling you, there is blood still in there. The catalase reaction showed positive for catalase. That is an enzyme produced by biology. Every bit of biology has that catalase. It reduces the oxygen, extra oxygens. Now, you see these little white particles? You may not be able to see them. Let me put a little more water on there. It dries up quick. All right, now let me hit it with the rag for a second here. Let it sit in there. You see that? Those are all little alveoli. And inside of those little alveoli, you can see all the little tiny little places where the air entered. And these popped, these exploded coming through space. That didn't erode into there. And you see these little white flecks. There's other places that have a lot more of them. Like up in this area up here. You see the little white flecks? Those are the ones that were all over the other lung, and they would have been all over this too. It's just that they cooked up. They're the weak spot. You see them? And there's alveoli all over this thing. I mean, it's a lung. It's just what they do. And I know there's blood in here. There's a lot of blood right there. That spot right there, blood just oozed out. There'll be areas where there's not quite so much blood, and there's areas that had a lot of blood in it that just bubbled right out, and that's one of the bubblers. She's a bubbler, baby. See it? Now, if I put catalase in there, I can almost guarantee you we're going to get a very good reaction. When I say catalase, no, I meant hydrogen peroxide. I'm going to let this dry up for a minute or two. And then, just let it dry up for a few minutes. And then I'm going to hit it with a little hydrogen peroxide. Yeah, we'll see what happens. Maybe nothing, but I think something is going to happen. This is TBD, to be determined. Okay, this is obviously blood. It's just taken forever to dry up, so I'm just going to do it the way it is. Now, that's the tube that runs in for the air pathways inside the lung. This is the pleura. This is that like dense coating that surrounds the lung, and we're down into the alveoli here. Now, these little white fabrics are these, I believe they call them plagioclases, those little white fabrics that are all over. Now, I'm going to put some hydrogen peroxide in there. Now, here's what happens. Inside of all living creatures, everything, plants, animals, everything, they have catalase. It's an enzyme. And that enzyme causes extra oxygen to be destroyed, removed. So if I put something in here that has a lot of extra oxygen, which is H2O2, it will kick off one of the oxygens and it'll bubble up into a bubble. Normally water, it doesn't bubble up, but you put H2O2 in there, it says, hey, there's an extra oxygen here. That thing is going to attach to something and cause us all kind of problems. Get it out of here. And they come over and cut it right off of the hydrogen, you know, the water, and they get rid of it. So we're going to see this bubble. But the bubbles are so tiny because you're dealing with individual oxygen molecules. So normally it just makes it like a fuzz. Sometimes they bubble up pretty good. Now, I'm going to have to come down close to it, otherwise we won't see the reaction. So hold on. All right, I'm up close. Let's get the lay of the land. Those are those little white plagioclase fibers, and that's the flora of the lung, all sort of melted down. And then we're into the blood. This is sort of just bloody runoff. And here we are at that deep channel that goes in to the lung. Now, let me focus down in there. You see way down in there? Look at that. Now, I'm going to load this up with hydrogen peroxide and we'll see where the bubbles come up. I may even have to get closer than this, believe it or not. Sometimes they're just tiny. It depends on how much blood is still in it. Don't forget, this came through space. This is not like something you picked up off the ground that was just laying around. Now, keep your eye on here. If you see white bubbles fuzzing up, it'll take a while. Shouldn't take too long, but it'll take a while. I think I'm seeing them coming up from the edge of the membrane, because that's where you see them a lot. We're seeing a bubble coming up there. Yep, she's going to start bubbling. There's a bubble. It's going to start cooking. Now don't forget, this came through space. Here she's coming now. Now they're coming out of here. It's got to penetrate way deep inside. That's what's going on now. And I just got to keep adding it until it really starts cooking. Now watch here, that's another one of the alveoli. And that goes way down inside. We should be able to get something seen out of there before too long. A lot of research is about waiting, my friends, and observing. Let's just keep watching over here. There's not a lot of it, that's for sure. But I'm seeing it coming out now. You see it? It's starting to eat its way deep enough in to where it's reacting. If this sits around for another, yeah, I can see them right now. You see them? They're starting to go good now. See it? See it? I'll bet you in about 5 minutes this is really going to start cooking, because I'm seeing them coming out of everywhere now. I hope you can see that. Yeah, it's starting to rock. Yeah, this is going to get going pretty good. I'm gonna do it up again a couple times and come back to this because it's starting to become reactive. It had to really penetrate. This, like I say, this thing had to be at least minimum 2,300 degrees, minimum, to turn into any kind of magnetic material. And it still has catalase in here. Catalase is a biological product. Now, I think it's all used up all the catalase, all the hydrogen peroxide. Let me put some more in, hold on. Okay, I dried it up pretty good. Let's go for round 2. See if we can get any good reaction this time. I would think sooner or later we're going to get something coming out of here fairly consistent. You can see it coming out. You see all that fuzz? That's the stuff. Let me see how close I can get here. A lot of this is reflection from the microscope, but a lot of it is also catalase reaction. You see this here? Just let's watch this for a while. That's what you expect to see from catalase reacting with hydrogen peroxide. And it's coming over here too. That's the exact reaction you would expect to see. There it is right there. So I know there's catalase in here. And if there's catalase, I would suspect that there's a possibility of having some kind of fragmented DNA. I'm sure it's not going to be good, because if this was really good, this would just be foaming, like bubbling like crazy. But it's not doing too bad. It's starting to get more and more. It's actually not too bad now. I just put more in there. Let's see if it has any effect. You can see it bubbling quite a bit. See all the bubbling going on? That's all catalase reactions. So this is still, and that's just on the outside edge. Now, if you cut right dead into the center, you would, I'm sure you're going to have some actually almost red blood, because I show meteorites all the time that have actually red blood inside of them, big ones, not mine. But I have a couple more meteorites here that also have, they're iron meteorites because they came through space. Just like this did. And this one here is clearly a lung. It has the Spurlock, it has everything on it. It has all the alveoli, it has the plagioclases, it has the pleura, it has the structure. It's a done deal. Okay, here's another lung. There's the Spurlock, and there's the little gnarly spot up in the back where they all have, where it lashes in. That little round spot. So that is the Spurlock. And the little white fibers are the fibers that we find on all of the lungs. There's some fibers here and there. Here's some alveoli. You see it? Basically the same as the other lung I just showed you. Let me put a little water on there. So this is also another lung. It's a little smaller than the first one I showed you that came through space. But guess what? So did this. This is an iron lung as well. It's a little tiny iron lung. There it is right there. All right, the big magnet here holds the big iron meteorite here holds the magnets pretty good. This one, no. It will hold it, but not well. All right, the reason is they smelt according to how much heat. And the heat is dictated by the size. The bigger they are, the more they smelt. That's why this one barely smelted. The other one smelted, better, but not a lot. And some of them coming through just turn into just a ball of iron. Okay, you see this here? That's an iron meteorite. When I say iron, it really means it's metal. All these different colors are from different crystals are from different types of metals, and they flocculate together and form these crystals. Now, there's an artery and there's a vein, and you say, "Oh, come on, Roger." I said, "Yes, it is." And this was big enough that it got so hot that it formed into an entire mass, which means it had to be above like 2,800, 2,900 degrees. At 2,200, 2,300 degrees, you get a semi-metal product. But there's some metal in here, and enough to harden up some of the metals, but not like this. This is a complete metal, and that means it got very, very hot. And here's what they look like right up close. There's the red blood. That's red blood. You can't deny that. That's as red gets. She said, "Don't get on no better redder than that." All right? It's just red blood. Red blood. And what do we got over here? Black blood. And all of these are different crystallizations because they're different types of metals. And they form different crystal colors, basically, because of their transition metals. So those two. Now look at this one. This is another meteorite. This was a gigantic, long meteorite. Basically like this. This came through space. That's the biggest iron meteorite on the Earth, I think. And it was a sacred object, and it was taken by us. Now, you see this little spot right there in the bottom? Remember I showed you in my other lungs how they had little spots that like tubes that went inside? Well, there it is right there, my friend. There it is. All right, that's a little alveoli entry, tube. And there's more of them in here, but that one there is just very, very apparent. But all of that is from iron or metals in the blood. And metals are just, it's everywhere in your blood. There's all those metals are in your blood. Virtually all the metals are in your blood. I was just looking at all the statistics of a study they did to examine 26 different metals. All the way up, they didn't even talk about iron, they talked about other ones, all the way up to uranium. And they're in your blood. They're supposed to be there. They're not there by accident. So there's a lot to think about here, and these things coming through space, that's biology. I don't care what anybody says anymore. I'm done worrying about people saying you're an idiot. This is a membrane. That is a membrane, 6 feet thick. That's how big these creatures were, 6 feet thick. 6 feet thick. In us, you'd hold that membrane up, you'd look through it, you wouldn't even hardly see it was there. They are very, 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 very thin. And this is what they look like. Right here. One layer here, one layer down here. In between is the fluid area, and the bottom area is just like this. Now, so this is literally only the bottom. This isn't both. This is just the bottom. The other one I showed you is the bottom and the top. Now let's look at it really carefully. This is the bottom layer, and this is the top layer. Anatomically exact. These are the ion channels. This little thing here, I think, is that little squirrely thing that comes up. Now, you see the edges of all this stuff? You see those edges? These are supposed to be sealed. This whole thing was sealed as one mat and couldn't penetrate through it. That's what a membrane does. People that get sick get loose junctions, they call it. And if the junctions open up like this, you can get penetrated through this membrane. It's a protective membrane. It's your immunity. It's the immunity of your body is in this membrane, basically. And in between, if it gets through the top, in here, there's supposed to be something that's going to kill it. And that's the fluid-filled highway is somewhere up in this area. And it attacks invaders, and it also, in the gut, in the intestines and so forth, it's secreted a whole bunch of enzymes and so forth to break down food. And then through the membrane, it has to say, okay, that's food, let it go through. Something else, it doesn't want to get through, it just says don't let it through. That's what a membrane does. It goes both directions. Stuff has to come in and it has to go out. And it has to secrete things, or somewhere there's a secretion of enzymes that are reacted when food comes in, that it has to break down. Carbohydrates, sugars, fats, all that different stuff is a different type of enzyme, which calls it from a different type of organ in your body. But it's also from a different type of bacteria. So you have to keep your bacteria in good shape. So this would have been sealed just as flat as could be. Well, not flat, but it would flap around and do all this stuff. But not a single thing could get through any one of these. Any one of those becomes a breach, and you get a little COVID thing coming through here, and it can get to that breach. You're infected. All right, and the same with any other pathogen coming through your body. It may miss that spot and just keep going, no problem. But it may find that spot, and then you got the issue. Okay, my friends, as I showed you, my research covers a lot of ground, and it shows a lot of things that nobody's ever taken into account before. And the things that in space that are coming through space, and the gigantic things that are on Earth, and the biology that is understood by researching the things that are here right in front of our face, needs to be taken into account. That's my last words on this for today. This is going to go a long time, trust me. This is all new. This is brand new. All the textbooks have to be rewritten. I mean literally all of them. History. Chemistry's changed. Space totally different than what they're talking about. It's saturated with particles. Light is slowing down. Physics is way off. I mean, just everything. There's nothing that is correct at the moment. And because of not understanding dipole electron flood theory and understanding that protons are made out of dipoles, they will never understand anything. So even though I'm talking today primarily about fascia, it all relates back down to the tiniest little particles that exist. You got to understand the whole play field. If you don't understand everything, then you're just picking one little thing and saying, oh, I understand this. No, you don't. Because what led to that? Whatever leads to everything is dipoles. Once you understand the dipoles, then you can work with whatever comes afterwards. And these are all made out of the same things you and I are made out of. Your eyeballs and your earlobes, your toenails, and this is all made of dipoles. Now, it appears to be the universe is a dipole. It's made of dipoles. They're a black and a white particle. Shown it thousands of times. And we have the light experiments to prove this. So until we start to get some exposure, this stuff is just unknown to the average person. The average person just dismisses it instantaneously. Instantaneously. Oh, the guy's crazy. Well, that's what's happened. And tell me, and trust me, this is all real stuff. So I'm going to leave it at that. Thank you. Love you all.